Okay, so hello everybody. It is my pleasure to welcome you to Virtual NGSymposium 2020. This is a one-of-a-kind event that marks the 5th year of our NGSchool history. And just at the beginning, I would like to give you a couple of technical notes and possibly some information about NGSchool and what the society is all about. And in the meantime, hopefully we'll have everything set up so that speakers can start with their amazing talks. All right. Right. I'm gonna wait for a confirmation that everybody can hear me, especially on YouTube. All right, I think it's fine. Great. So as I mentioned, this year marks the 5th anniversary of the 5th edition of NGSchool. Obviously, this year is a bit different. The first NGSchool, the first NGSchool, the NGSchool began in 2016, and it started just as the summer school. The idea was to bring an excellent training that is affordable to the trainees, especially to trainees from Eastern Europe, and where most of us organizers come from, and where we found that there was really no computational biology and bioinformatics training that was available for us back then. So it started just as a summer school, but it evolved in something so much more. It evolved into the community of people who are working together to on the idea to pay it forward. Most of us, including me, are former participants of NGSchool summer schools or conferences who decided to help and organize something for our benefit and for yours benefit, for benefit of the trainees, and bringing an excellent training to people who otherwise couldn't be able to go through it. In 2016, we started with just general NGS data analysis school because this was what was on demand at the time, and there wasn't anything that could bring us this type of training. In 2017, we came back with even more participants, focusing this time on single-cell sequencing. In 2018, we had the first school that was both computational and wet lab. We focused on nanopore sequencing and personalized medicine, and we worked together in parallel sessions during the hackathons. And some of the hackathons even resulted in participants getting publications after that. We published 2 publications with new genomes that were sequenced or assembled during the conference. And last year, we had a special fall edition of our school, focusing solely on computational part, on machine learning for biomedicine. And even though it was fall and the weather was so much worse, we were very happy to meet um, and work on different aspects of machine learning for biomedicine and more. This year also marks the first time that we had talks that were recorded and then released to the public. Also, there was an live, online live discussion panel. That was our test to. Sorry if somebody hears noise. I'm sorry for this technical issues. Yes, so last year we had recorded talks and online discussion. This was a test, and I think we passed because and this is how we were a bit more prepared for what was to come. So for this year activities, which mostly were held online. In general, obviously the world had to adjust to global pandemic that is going on right now. During late May or June, we agreed that there is no possibility of holding the school in person even later this year, based on all of the data. However, we decided that we are not going to let this pandemic win, and we're still going to do our best to bring the training to people who are in need of it. So we decided to hold a session. We decided to hold a series of sessions in NG seminars that touched different areas that will be helpful to trainees. We had 13 tutorials and workshops that are available on YouTube right now. And the topics vary from introduction to programming to data science, data visualization, and even isoform detection from long-read RNA, RNA-seq. And we are very happy and we are very humbled to receive continuous positive response to our outreach, even though we are living in these troubling times. And now we are holding this NGSymposium, which for sure is not equivalent to our stationary summer schools. However, we are very happy to be holding to prepare for you exceptional talks, and I'm very happy that I am able to do this introduction and tell you a bit more about what is to come. So we have 3 keynote speakers. First keynote talks is going to happen in just a couple of minutes right here. Christopher Yao is going to tell us more about data models, complexity, and what to do about it. He's a professor of artificial intelligence at the University of Manchester and a fellow at Alan Turing Institute. And I'm not going to spoil anything, but we are very excited to be able to listen to this talk. Tomorrow, there will be 2 more keynote talks starting at 3 p.m. Central European Time. The first keynote talk is going to be given by Akrosh Ratan, who is assistant professor at University of Virginia, Public Health Sciences and Cancer Center. His research is focusing on studying genomic variation and genetic diversity and its consequences on species health and survival. Next, at the end of the conference, we're going to have the last keynote session. Rosa Bento Tormo from Wellcome Sanger Institute is going to tell us more about temporal and spatial dynamics of human endometrium. Her group is aiming to understand, to further our understanding of adaptation of immune cells and tissue and their function in steady state and inflammation. These talks will be held at 5:45 p.m. Central European Time tomorrow. Just like I mentioned, some of us here in NGSchool Society were participants before, and we are very humbled by the by the enthusiasm we are getting from you and participation in our events. During our sessions, we're going to present. There's going to be a coffee break. There's going to be a possibility for you to find out more about our NGSchool Society. So if you are watching this on YouTube and you have not yet joined our Discord, please do so using this link. And when you go to the Discord, you can fully participate in the conference. There are channels that allow us to discuss sessions as they are taking place. You can ask questions in Discord channels and on YouTube. And of course, network during the coffee breaks. Yes, so this is a session schedule for today. It looks very exciting. There's going to be 2 sessions with 1 keynote speaker in a moment. And then tomorrow, there's going to be 2 more sessions, and at the end of the conference, we're going to tell you a bit more about our society. I'm very happy to be here, and I'm very happy to be able to listen to these exceptional talks. And if you have never heard about our society, welcome. And if you have heard about this, please enjoy. I think we are ready for the first speaker now. The chairperson for the first session is going to be Lidia Lipińska-Zubrycka. Lidia, are you here?
Yes, I'm here. Can you hear me? Okay, I think it's fine. So hello everyone. My name is Lidia Lipińska-Zubrycka from the Institute of Genetics and Biotechnology. I do my postdoc at the University of Warsaw, and today I have a great pleasure to open first session of our NGS symposium. Our first special guest is Christopher Yao, Professor Christopher Yao from the University of Manchester, and first keynote lecture, Data Models Complexity and What to Do About It. Chris, the floor is yours.
Hi, can you hear me?
Yes, we can hear you.
Great. Can you see my slides though?
Yes, we can see your slides. Everything is fine.
Great. Thanks for inviting me to talk, everyone, and thanks to everyone tuning in. I'm just moving a few windows around whilst I get sorted. So thanks for inviting me to talk. I gave a rather mysterious talk title, but what I really want to talk about is some of the research the now former student of mine, Jiwon at Oxford, did as part of her PhD. She's now finished and is doing a postdoc as part of her PhD, and a modeling problem involved in that research, which posed some kind of interesting kind of statistical issues. And what I'm going to do in this talk is present that problem to you, the way we kind of tackled that problem, and perhaps a bit more unusually, to take you on a little bit of a deep dive into how we solved that problem, more so than might be presented in a kind of normal kind of a research talk or in a research paper, to really get into the real details about the statistical concepts and ideas that were behind our thinking when we tackled this issue. So just by way of introduction, I'm a professor of artificial intelligence at the University of Manchester, and my main kind of interests are in statistical machine learning and the design of different types of algorithms and methodologies to tackle problems in biomedical sciences. But many of the people I train are not only statisticians and machine learning people, but also have people who work in the wet lab, and G1 was one of them. And so one of the things I like to do with students who don't have the same background to me is to make sure they have training in appropriate statistical concepts, so that even though they may not necessarily have the experience or the ability to apply these in a rigorous mathematical ways, they're able to reuse existing tools in a way that allows them to solve real world problems. And in this talk, what I'm going to be talking to you about is a problem we sort of encountered when we did some single cell sequencing of real world cancer patients. When we then proposed a solution to that problem using an approach which, if you present it just as it is, has no real obvious statistical foundations. But what I will reveal in this talk is that actually there was a lot of statistical thinking behind it and its underpinnings, and that really what we took was a kind of pragmatic approach of incorporating a statistical mindset into solving this problem without necessarily having to do a deep dive into mathematical modeling. So just to give you some context, what I'm interested in is cancer, and one of my kind of main research interests is in ovarian cancer, in particular high-grade serous ovarian cancer. And high-grade serous ovarian cancer is a particularly challenging disease. It often presents in women at a very late stage, by which time it has metastasized and often is spread across the body from the abdomen. It's a disease which is incredibly difficult to treat because it's diagnosed late, and survival rates and prognosis for high-grade serous ovarian cancer patients is very, very poor. In fact, over the last kind of 20 to 30 years, we haven't been able to drastically improve the survival rates for patients with this particular type of cancer. Now, to tackle this disease, what many people have done is used genomics and omics in general to profile lots of ovarian cancers, look at how those cancers have genetically or molecularly evolved, try and understand the dynamics of the disease, how it reacts under different types of treatment. A lot of data science and computational work has gone into these types of studies, including, for example, the Cancer Genome Atlas or the ICGC studies. What we're particularly interested in is a different side of ovarian cancer, which is its origins. So there's been a body of evidence over the last 10 to 15 years, which has indicated that actually ovarian cancer is slightly misnamed. Actually, the initiation site for ovarian cancer is very likely to be in the fallopian tube, and in particular, cells which are in the fallopian tube epithelium. And so there's a body of evidence which says that the cancer or early initiating cancer events happen in the fallopian tube epithelium. These kind of grow and then spread out from that site of origin. And so what we're particularly interested in was characterizing the population of cell types which sort of comprise that fallopian tube epithelium, characterize those cellular phenotypes, and to see to what extent those cellular phenotypes in the fallopian tube epithelium are associated with cellular phenotypes in the tumor. So to set about doing this, what we did was to start doing some single-cell RNA sequencing of fallopian tube cells, which we took from cancer patients, in this case ovarian and also endometrial, which are kind of related, but also later on as well, patients which didn't have cancer but may have had other conditions which required surgical removal of their fallopian tubes. And one of the things that we had to work out early on was how we were going to do this kind of experiment, and how we were going to, in particular, preserve the cells before we put them through single-cell sequencing. So one way, and perhaps the most logistically laborious way, is to take fresh cells almost immediately from surgery, get them processed in the lab, and straight into single-cell sequencing, so that we get data, as fresh as possible from the patient. This has obviously some practical issues because surgery can take an unspecified amount of time. We have to potentially wait for all hours, waiting for the patient and their samples to be ready. What we'd ideally like to do is to either freeze the samples or to leave those samples in culture so that we can then, at appropriate time, pull them together and then sequence them when things are convenient. So what we did in this experiment was to collect samples and preserve them in these 3 different ways, single-cell sequence them, and then to look at the expression of these cells under these different preservation regimes to see what the effect was of each of these preservation methods. And in the sort of bottom left-hand corner, you can see a principal component analysis plot of those cells under each of these 3 conditions overlaid on each other. Actually, I've taken out the frozen cells, so they're only culture cells overnight and culture cells which have been left for 2 or 6 days. What we see is that there's a pool of cells which are fresh cells to the left of the bottom left-hand diagram. But if we then look at the cells which were cultured, those actually occupied a different position on the PCA plot, meaning their expression profiles were fundamentally quite different. And then if we left those cells in culture for even longer, those cells actually split and generated 2 different cellular expression phenotypes and moved to a different profile altogether. Now, this kind of disturbed us, but wasn't entirely unexpected. If you freeze cells, you will stress the cells, and those will probably be replicated in their expression profiles. If you culture cells in a different environment from which they are normally located, then of course, over time, the cellular phenotypes might evolve to exhibit a different kind of profile altogether. All in all, this meant that we were a bit worried, and so to be conservative, we focused the rest of our analysis only on fresh cells which were derived from these patients. So what we did was to focus in particular on the set of fresh secretory cells. And we know how to identify those secretory cells by looking for a couple of markers, in this case KRT7 and EBCAM, and the absence of expressions of a couple of other marker genes to pick out that group of secretory cells. We also excluded any potential cancer cells that might have migrated into the fallopian tube by looking at copy number variants or apparent loss of heterozygosity using the RNA-seq data. But what's kind of immediately obvious from that UMAP plot on the left is that we have a bit of a problem. So remember, we are taking the cells fresh from the patient who's undergoing surgery directly into a lab. So in this case, a batch is also a patient, and we need to process that batch and therefore that patient immediately. And so there's a confounding here. If there are any patient-specific effects or batch effects, these are going to be mixed up and completely confounded. So in that UMAP plot, you can see that actually, if you color the cells by a patient sample or patient, what you find is that there are groups of cells which group together by patient and are probably going to obscure any cellular subtype effect. So to overcome this, what we did was to develop a meta-clustering approach called ClinCluster. And the idea of claim cluster was to do three things. In the first step, we would take each patient individually, and then would cluster the cells taken from that patient to identify any subtypes. Then what we would do in the second step is to take pairs of clusters across two different patients and measure the dissimilarity or similarity between those clusters. And the way we're going to measure similarity is via differential expression analysis. So we're going to count the number of differentially expressed genes across these two pairs of clusters that we're comparing, and to add those up and to derive a score of similarity based on the number of differentially expressed genes. And then in the final step, once we've derived these scores, we're going to use them to finally merge clusters across patients to come up with a final set of cellular subtypes. So if we do this, what we find, and we apply this to our data, what we found is that there were at least 6 secretory cell types in the fallopian tube epithelium. And each of these we eventually were able to kind of identify by looking at the expression of marker genes. So we found some cell type, which is inflammatory cell type, cell cycle cell type, a differentiated cell type, and an EMT cell type. And on the right-hand side here on the heat map, what you see along the bottom is that there are multiple clusters consisting of multiple clusters. And each of these clusters consist of multiple patient cells. And the important thing here is that the results suggest that our algorithm, clustering algorithm, has seemed to work. Each cluster is not simply a function of one particular patient. So the claim here is that this method allows us to correct for confounded batch patient effects when looking for distinct cell types. And the question is whether really this is true, and how we might go about answering this question using statistical thinking. So when I try to tackle a modeling problem and to answer questions like this, I usually ask myself the following 3 questions. 1, what is the mechanism by which the data is generated? What is the model design that I should adopt? Is this the only possible mechanism? So it is a model choice problem. And how do I evaluate which model is most appropriate? Is there a model selection problem here? And with these 3 kind of thoughts running through my head, then I tackle each of the statistical issues that are associated with a problem one by one.
I'm sorry to problem with your slides. We can't see your slides. Wait a moment.
Oh.
Someone, can you help us with the slides of Chris?
Sorry, my browser window seemed to have crashed.
Okay, so we will wait for a while. Okay, now it's fine. Sorry, and thank you.
I'll go back a bit, just in case you missed anything. Okay. So I'll go back a few slides. And so the claim here on the left is that we've developed this meta-clustering algorithm, which is supposedly helping us to correct for confounded patient batch effects when we're looking for these distinct potential subtypes. And the question is, no. Unfortunately, that tab just keeps crashing on me.
Now it's fine. Again.
Okay. So the idea is that we have this meta-clustering algorithm. It's supposed to solve this confounded patient batch effect problem, whilst simultaneously allowing us to look for distinct cell types. And the question is that it's seemingly this algorithm is constructed through a series of 3 independent steps. What is the underlying theoretical or methodological framework that makes those 3 steps seem sensible? And how might we go about answering this question as to whether this method is actually a well-founded approach for tackling this problem? So when tackling a modeling problem like this, I usually ask 3 questions. One is, what is the mechanism by which the data is generated? So what is the model design which is most appropriate for analyzing this particular problem? Is this the only possible mechanism? Is this the only model which is appropriate, the only model choice I can make? And if there's not, and if there's multiple models that I could apply, how do I evaluate which model is most appropriate? How can I do a model selection?
I'm sorry, we have the same problem. I'm really sorry for this technical issue. We'll try to do something with that.
Seems to just keep crashing on my tab.
Could you close some tabs? Maybe it's a problem, too many things in the same time.
Okay.
And now we can see your slides. Maybe we will continue, and if everything will be not good, we will do something more.
Okay. So let's move on to looking at this particular problem that we're trying to tackle, and this is a kind of cartoon illustrating the problem. So the problem we have here is that we have different batches, but those batches are also different patients as well. And we want to identify different cell types. And within each patient and batch, we have different genes. Here I just showed gene 1 and G2. And we might sort of break down the expression related to each of those genes into 4 different kind of components. One which is a component which is related to patient and batch. The second, which is related to the gene-specific effect, the degree of expression, which is purely due to that particular gene. And then a component of expression, which is related to the cell type that gene is sort of linked, associated to with. Now, if I look at those same genes in a different patient, the magnitude of expression might be slightly different, because the background patient and batch effect might be is potentially different, but we might suppose that the cell type and the gene effects will be consistent across these different patients and batches. So, given this framework, why is it that this meta clustering algorithm seem to work? Why does it seem to work? So, if we take step 1 of this algorithm, what are we doing? We're taking the expression or count matrix from these cells and across all these genes from our single cell sequencing experiment. And then what we're going to do is take each individual patient at a time. So we're going to take a subset of those cells belonging to each patient, and then to cluster those cells within that patient to identify different subtypes. And in the context of our kind of overall framework for analyzing this data, this is a useful thing to do. So if I effectively take each patient's cells one at a time, what I'm effectively removing from my consideration is the patient and batch effect from this particular problem. So I can effectively control for patient and batch effect by just looking at one patient at a time. I can also somehow somewhat remove the effect of the gene-specific component, because now I'm going to be clustering cells. I'm going to be aggregating information across all sorts of different genes. So I'm going to remove the specific gene contribution to my variability by simply pooling that information together when I do my clustering. So clustering within a particular patient is useful because now I don't have to worry about the patient and batch effect. I'm going to aggregate all the gene-specific information and purely within that patient be able to derive the cell types. And I can do that for each batch at a time. But the choice of clustering method is an issue here. And what we did was to essentially do a simulation study where we used a piece of software, a single cell expression simulator called Splatter, to generate a number of conditions with variable genes and variable number of batches, and then applied various 4 different sort of clustering methods to determine which of these 4 clustering methods might be most useful for this first step of our algorithm. And we looked at various things like affinity propagation, or k-means, or k-nearest neighbor. But the thing that we eventually settled on was spectral clustering, because it appears to do well under all the scenarios we considered. So this in some ways kind of wraps up thinking back to those 3 questions I asked. We did the model, we thought about model design. So what is it we're trying to do? We're trying to do clustering within a particular patient. We needed to think about model choice. There are different ways of clustering, so we took that into account. And then we selected a particular clustering algorithm based on an evaluation framework, which involves lots of simulations, lots of different conditions, and in this case, using a metric, the adjusted RAND index, to select which clustering method was most appropriate. But this spectral clustering can only be used for step 1. We can't finish here, because if we apply spectral clustering across all the data sets, it would be highly influenced by the batch effects. And this particular simulated example illustrates what I mean by that. So in this simulated example, there are actually 4 cell types, designated by group 1 to group 4. There are actually 3 batches as well within that simulation, but batches 1 to 3. And if you apply spectral clustering directly onto this, ignoring the batch effects, what you'll find is that it will cluster the data, the cells, into different groups. But those groups could either be actually real cell types, or it could be some amalgamation of both batch and cell type. So spectral clustering on its own is not enough to solve our problem, but it's useful to look at cell types within a particular patient. So the next step of our algorithm is that now we've constructed these cell types within each individual patient. What we're going to do is start looking at across patients and start comparing pairs of clusters to work out if they're similar or dissimilar. And if they're similar, what we're going to be saying is that, they're very likely to be actually functionally the same, and therefore this cluster in one patient is actually the same cell type as another cluster in a different patient. And what we'd like to do is to compare all possible pairs of clusters across these patients to construct a distance matrix that we can then use for merging in step 3. So going back to our kind of background framework, what is this step really trying to do? Well, in this particular step, we've already worked out the cell types from step 1. We've used spectral clustering to delineate the cell types. So what we can do is, because we're looking at pairs of cell types across patients, we're conditioning on cell type. So we're kind of removing cell type from our consideration when we're looking at these 4 components of the expression. We can now correct or regress out patient and batch effects. And then we can look also at the gene-specific effects within this framework. So because we've conditioned on which cell types we're going to be looking at within each patient, we can remove that component from consideration and then purely look at the gene component, regressing out the patient and batch effects. And what we're going to be doing in particular is a differential expression analysis to look at which genes are actually differentially expressed between these different groups of clusters. And so, of course, there are different types of differential expression analysis which are available to us. In this case, we used EdgeR and did two different settings within the limma package. And in fact, all of these differential expression methods recognize the structure underlying our initial clusters across the different batches to the same degree, more or less. This is in a simulation study, this is not surprising because the clusters are relatively clean. So we decided at this stage not to choose any particular differential expression analysis tool, and actually to take both of these packages through to the next stage for further consideration. So in the final step, what we're now trying to do is merge all the things that we've discovered in the previous two steps together. And so what we are interested in is taking that distance matrix that we've computed in step 2 and using that as a basis to merge the initial clusters that came out of spectral clustering on individual patients, and to create these final set of clusters, which should hopefully represent a particular cell type, but from which the expression value cells that we've captured by each of those clusters are actually derived from different patients. And then going back to our sort of overall statistical framework, why are we able to do this now? Well, basically in the previous two steps, what we've been able to essentially do now is to essentially remove out the nuisance artifacts that we're not really interested in. So patient and batch effects have effectively been regressed out in step 2, and we're able to kind of aggregate out the gene effect, specific gene effect, when we compute this overall score of cluster similarity. So now all that's left in step 3 is just to merge the clusters based on the cell type information only. And so given this kind of distance-based score, a very straightforward way to sort of cluster based on that distance matrix is to use some hierarchical clustering. But of course, we didn't decide on which differential expression analysis method we would use in step 2. So we actually looked across the different distance matrix, different distance matrices across our 3 methods, and did the hierarchical clustering in each case. In all 3 examples, actually the final clustering merging gave more or less the same kind of output. And so really in practical terms, the choice of differential expression analysis that you would use in step 2 is going to be more influenced by your personal preference and probably also computational speed. And if I recall correctly, limma was a bit faster than EdgeR for this particular purpose. So to go back to the original question of whether this meta-clustering algorithm is methodologically well-founded, and the answer is sort of yes. And it's well-founded, though, assuming that we have a linear additive structure. So implicit to that kind of overall statistical framework I've been talking about is that the expression for any particular gene in any particular cell is going to be equal to a cell type specific factor, a gene specific factor, a patient, and a batch factor. And in this particular experiment, patient and batch are kind of confounded. And so we use these 3 sort of separate steps to allow us to learn something about the cell type factor, which we're interested in, whilst at the same time not overcoming the fact that patient and batch effects are confounded. So in step 1, we kept things constant by working within batch and then aggregating across genes to allow us to learn something about the cell type within a particular patient. Then in step 2, we conditioned on cell type and regressed out patient and batch effects using our differential expression analysis tools to learn something about the individual gene effect. And now that we've learned something about the gene effect and are able to regress out the batch and the patient effect, we're finally in step 3, able to learn, aggregate our information about cell type across all the patients. And so this gives us a lot of confidence when we're making comparisons against external methods, that the performance and the sort of positive results that we see in our experiments are not due purely to a set of simulations which perhaps strongly favor our particular method, or that we've implemented things in a way which are sort of more efficient or better than others, but there's actually a firm statistical foundation behind our method, which explains why our performance is good. So in this particular simulation study, again, we use splatter to generate lots of different scenarios, different number of cell types, different number of batches, and we compared a range of methodologies which attempt to look for different cell types from single cell expression data, but correct for batch effects. But in most of the other methods, they only correct for a single type of batch effect, rather than this unusual situation we have here, where we have confounded patient and batch effects. So what we simulated was this confounded patient batch effect problem, but most of the methods we compare against would only be able to handle one of those dimensions at a time. And what we show here is that this CLIN cluster algorithm, because of the way it's designed, is able to handle these simulated situations relatively well, whereas some of the other methods have variable performance depending on the particular scenario. And so when we sort of aggregate the information across these different scenarios, what we found is that the average kind of clustering performance of our method is strong and very good compared to the sort of ground truth that's used in these simulations. But more importantly as well is that the stability of our method is very strong as well. We're very consistent given the same scenarios in the clustering performance. What we found in other methods is that even when they were clustering well, sometimes using a different data set under a slightly different scenarios, they would then give very poor performance. And the advantage of our algorithm, because it's kind of has this background sort of statistical thinking behind it, is that if it doesn't do well, we know what the underlying assumptions are that we need to go back to address, in this case, the linear additivity and the various implementations we use to implement each of those steps. With some of the other methods, there isn't necessarily an underlying statistical kind of framework behind them. So it's very difficult when performance is unstable to understand exactly what is going on. So we didn't just look at simulated data, we also looked at a range of real-world single cell data sets. Of course, with actual single cells data set, we won't necessarily have ground truth true cell type labels, but we managed to find a few data sets, and this one is from a published melanoma paper, where we used the curated annotated labels that they created as part of their study as our kind of ground truth. So this is not technically a real ground truth, but we're going to try and see if we can recover the annotated labels that were published as part of this particular study. And what we found here is that with CLIN cluster, what we were able to do in figure C is that we were able to recapitulate the cell populations that were identified in the original studies. That's figure B here. Whereas the other methods, which aren't able to account for the sort of combination of patient and batch effects, had variable performance. So the only other method which did reasonably well in this particular setting was a piece of software called Race ID in this case, which did fairly well, and so it's very comparable in performance to what we had. So I'm going to end here by skipping a few slides because my battery is actually about to run out. But what I've hoped to show you in this kind of slightly interrupted talk is a solution to a problem which we implemented using an apparently myriad of methods and heuristics, but were actually underpinned by a lot of statistical thinking. Now, if you're a purist working in this kind of domain, you would probably just say, actually, since you've done all this statistical thinking anyway, and you have this linear additive model, why didn't you just build a probabilistic model in the first place and do formal statistical inference based on that? But this type of approach requires time, requires experience, and requires a particular expertise in being able to implement those probabilistic modeling ideas. I love that type of approach. That's kind of my main area of research and what I kind of do. But I also recognize that everyone has limits on their time and resources. And so a much more pragmatic and sort of more general approach to tackling these type of problems is actually to understand these statistical underpinnings and the concepts and ideas that might underlie these problems, as we did with the development of ClinCluster, but then to actually reuse tools that are already available to us. So in this case, we reused spectral clustering, differential expression analysis tools, and hierarchical clustering, but to use them in appropriate and sensible ways with a background, with that statistical framework in the background. And here, for the development of our particular algorithm, we were able to piece these things together in a way that gave us the type of analysis results that we were interested in, and we didn't really have to do any kind of hardcore statistical modeling or mathematics in it to enable us to do that. So I'm just going to end by thanking my student, Jiwon, who did most of this work, and also her co-supervisor, Professor Ahmed, who's based at the Ovarian Cancer Lab in Oxford, and who sort of led the single-cell sequencing study. These are the various people who funded the research, myself, Jiwon, and Ahmed, and I'm going to save lots of time for questions.
Thank you very much for interesting talk. We have some questions for you. First question from Anna Maria. Some comment and question. Interesting talk. It's very exciting work. Did you look at what I have to do it bigger for me? Okay. Did you look at what are the genes? I'm sorry, it's my first time as a chairperson.
It's okay. I don't even know which window I'm supposed to be looking at the moment to see the question.
Okay, it's on Discord. Did you look at what are the genes that drive this finite clustering, and can they be used in sort of predictive manner? It's in the left panel of Discord. Sessions discussion. I want to read it.
Yeah, so it will be in the paper. It's in Cancer Cell, which has been published. I can't remember the details of it now, but we did look at the set of genes which were driving some of these cell types. And actually, I think I showed on one of the earlier graphs a heat map showing some of those genes that were driving individual subtypes. I think it's this one. But the detailed information about particular genes and how they might be used potentially in predictive models in the research paper if you're interested in looking at further.
Kasia asked, I would be very curious to learn how to visualize the data. And at all, do you perform some kind of dimensionality reduction?
First of all, how I visualize the data? Well, we're in a single cell world, so most of us are using combinations of either t-SNE visualization or UMAP visualization. Depending on which camp you're in, you might use one exclusively over the other, but we're not in either camp, so we'll use both, and you'll probably see some of the figures actually show both in terms of visualization. For this particular problem, we weren't doing any explicit dimensionality reduction for the clustering. We were using spectral clustering, which is itself a method that incorporates dimensionality reduction as well as doing the clustering. So if you're interested, obviously look up what spectral clustering does. Often with some of these things, we might also do an initial principal component analysis reduction just to remove any upfront linear effects to make things like spectral clustering or nonlinear dimensionality reduction techniques work a little bit faster. So with some of the more nonlinear techniques, if you put in like tens of thousands of genes and ask it to do dimensionality reduction, it'll probably do it, but it might take a long time to do.
Okay, thank you. And next question from me. It's my question. Yeah. What do you think are the most important challenges for the next several years in machine learning in the field of human healthcare? Because you are a specialist, and
yeah,
I'm sure you have some ideas, some challenges.
There are a few sort of generic open challenges, and I think the talk after me will be addressing one of them. And one is using sort of heterogeneous data sets and how we integrate them. And this is an area I could have also given a talk on this as well, with the same kind of motivation. Integrating data sets is across different like omics, for example, is really difficult. I always encourage people in my group to kind of step back and think about it carefully, because sometimes you could be in a situation where you're trying to integrate two data sets where actually, if you step back, there's no reason why you would want to integrate them. They might be looking at completely biologically orthogonal things, and therefore there's no value in integrating them. Or if there are sort of commonalities they kind of target, they use different data sets, you'll need to kind of focus on what it is that's common and kind of model that. So integrating data is definitely one area that's going to be important in future years. The other kind of major areas, kind of more general things, which is scaling and having to deal with more data, more samples, more cells. And obviously, we're in a strange situation now where the rate at which we're able potentially to acquire data, particularly in a single cell world, is almost going up exponentially, and our ability to kind of tackle that information is probably not going up exponentially. So scale is a particular issue. And part of that, related to that, is actually perhaps from a computational point of view, having to upskill to take advantage of new modern sort of programming or development frameworks that can handle that. So many of you will know that increasingly more people are using frameworks like TensorFlow, Google's TensorFlow, to do computational bioinformatics problems. And that's because Google have, within their TensorFlow framework, taken care of a lot of the issues related to kind of scalability and doing that kind of automatically in the background. So scalability and also the skills required to access tools which enable you to scale methods easily is going to be, I think, a big challenge. And probably one final thing is that if you're looking at human health as well, bringing in clinical information from electronic health records, clinical phenotypes into our studies is also going to be interesting as a particular type of data integration. You can think of electronic health records as just another data modality. But bringing those in is going to be quite important in some sense to guide our analyses so that we're actually doing identifying subgroups of patients or subgroups of cells which might actually influence outcomes and endpoints, which in the clinical world is how you would actually judge the efficacy of any kind of treatment or intervention.
Okay. Question. We have time for some questions still. So next question from YouTube, from Michał Krasowski. The approach discussed appears very well suited for discrete covariates. Would it be possible to easily include a continuous covariate, let's say age of a patient? Yes,
absolutely. And I think this, I mean, as it stands at the moment, you probably wouldn't be able to do it because some of those steps would, particularly the sort of clustering step, would assume implicitly that the covariates are discrete. But because you've got this background framework of the underlying statistical model, you would just need to say, actually, the variable of interest that I want to include is continuous, and then I would just need to swap in a method which is appropriate for continuous covariates into there. So you might be able to reuse some of the steps that I've discussed, but you may need to modify one of those steps to include a method that deals with continuous covariates.
Okay. And last question. Possible to find some golden rule to design the best treatment for cancer from your point of view, or does every patient require personalized therapy?
Sound was a bit noisy, but I think I heard the question correctly. I think what's the best way of treating patients, or is it personalized? It varies obviously depending on the cancer type. Certainly with high-grade serous ovarian cancer, because it presents so late, it's extremely difficult to treat. Most of the patients relapse within 2 years. The standard chemotherapies don't work. We're not in a regime, despite our desires, where personalization, we're ready for personalization yet. And one of the things that I do in my research group, and there are many others around the world, is something to look at genomic evolution or evolution of cancer within patients. And we've done studies where we've looked at genomes or molecular profiles of ovarian cancer patients, and we see differences not only between patients, but sometimes even within different sites of tumor sites within the same kind of patient. So the disease is evolving constantly, and there are differential kind of pathways. So personalization, it's coming, and I think it's a goal that we should aim for. But for certain types of cancer, we're a long way from that yet, because actually, without these molecular profiling studies, we don't really know enough about how the disease evolves to be able to target in a personalized way things that might affect outcome.
Okay, we have one more. It's general question. How can big data help us to treatment of cancer?
I think I heard that as how can big data help us with cancer, and I think it goes back to a bit to my previous answer, which is that cancer is a very heterogeneous, constantly evolving disease, and so the data requirements are even higher than in the study of more common diseases or rare diseases. So if you're looking at a sort of rare genetic disorder, all you'd need perhaps potentially is just their germline DNA sequence, and that would be the only data point you need. In a cancer, because the disease is constantly evolving, if I took a tumor sample now, sequenced it, and then waited 3 months later, took another sample and then sequenced it, what I'll find is that there are changes, there are new somatic mutations, and new chromosomal abnormalities. And so even for one patient, and it's already, there's already evidence to suggest that actually it's useful to take multiple samples, both from within the patient at the same time, but also to follow those patients over time as well, do a longitudinal sampling, take a longitudinal sampling approach to collect data to watch their disease evolve. And so some of the studies which look at things like circulating DNA or ctDNA are a way to kind of use omics to kind of monitor the evolution of the disease. And the downside and the upside of that, depending on your perspective, is that we're going to need, this leads then to collecting more data to enable us to really characterize what the dynamics of the cancer are.
Okay, thank you very much. I think it was the last questions. It was our great honor to invite you for our NGS symposium, and we are very happy that you decided to have a talk. Thank you very much for excellent talk, for answers of all the questions. Thank you.
Thank you.
second part, the next part of our session will be a talk of Karolina Sienkiewicz. Karolina Sienkiewicz is a software engineer at Center of Public Health Genomics at the University of Virginia. She is also main organizer of this NGS symposium, Virtual NGS Symposium, and one of the organizers of NGS seminars and NGSchool. We are happy, Karolina, to hear your talk about data integration and molecular subtyping of multi-omics data. The floor is yours.
Thank you. I hope that you can hear me, and I hope that there will be no more interruptions as they were at the introduction. So I'm going to talk more about the data integration and molecular subtyping of multi-omic data. So let's start. Okay, so when we are talking about multi-omic data, about molecular subtyping in particular, we're basically thinking about a clustering case. We have samples, and we want to find samples that are more similar to each other in the molecular sense than to samples from other groups. So basically, we have data, for example, gene expression, and we want to find groups in our data or clusters. And these clusters, why do we want to identify groups at all? So basically, these groups may have an underlying molecular mechanisms that result in differences in, for example, expression of some genes, some biomarkers that may result in different answer to a treatment. So it may be very important for us to identify which group the patients belongs to, so that we can sufficiently prepare ourselves to and select the best treatment. Right now, we have, with the development of high throughput genomics, we have a lot of multi-omic data, so a data of different types, of different levels, and more often than not, we want to integrate data of totally different types. For example, when we are talking about multi-omic data, let's each omic would be a different data type. So let's say we have a methylation and expression. So these are two omics, and we want to combine this data. And for example, we want to combine it with some even different types of data, like somatic mutations or copy number variations. And by integrating this data, we gain the opportunity to discover a system-level insights that may be not possible to discover when just looking at each data type separately. So this is a very exciting area of study of how to efficiently integrate this type of data together. And there's a bunch of different approaches to this problem. So here's a very good figure that describes these approaches, the most common approaches. So we have either early or late integration, by which I mean in early integration, we concatenate all of the omics together, and then on this big data set, we use our standard clustering methods that would be useful for single omics. And this is an approach that seems promising because you can use the algorithm that you already have. However, there's an issue of normalization, that in general, the omics that have a higher number of features would be favorized over the omics with a smaller number of features. So we need to think carefully about how to normalize our data. And also here we totally ignore that our data from different omics may have completely different distribution. In late omic, we want to cluster omics separately. And then, this has the advantage of using an omics-specific method for each of the omics, for each of the data types. But then we have to attempt to integrate these clusterings together, which may be very hard. Then we have a similarity-based methods where we compute similarities per omics. So basically we are looking at samples and we find samples that are similar, that we calculate similarity of samples to each other per data types, data type specific similarities. And then we integrate these similarities together. So here, the advantage here is that so for most of the multi-omic data sets, we will have biological data sets, we will have much more features than samples. So if we are working on similarities, these approaches will be much quicker. However, we have to think carefully about the interpretation, because right now, if we calculate similarities, then we stopped working on features directly, and we are working on similarities. All right, then there are dimension reduction methods where we attempt to find a low dimension representation of our data. So very, very frequently we assume that the data has a low dimensional representation, with a dimension, the low dimension being very frequently the number of clusters that we are looking for. And then we are clustering this new representation that we achieve. And there are also statistical methods, most notably probabilistic modeling, which tends to be very specific. So the models tend to be very specific towards data types. So a model for somatic mutation may not be the same model that you would use for expression. What are the main challenges? So other than the approach-specific challenges, we have the challenge of missing data. So in biological data, more often than not, we will have a lot of missing data, not only missing features, but let's say we have expression and methylation. We may not have the methylation, for example, data for some samples for which we have expression, but we want to include this data in our analysis. So this is also a missing data case. And the last thing would be stability. So we want to find a method that will give us stable results for when we want multiple times, even if it's not always fully deterministic. Then we want to find a stable solution, stable amount of clusters, stable groups, because we want to build our biological analysis based on these groups. So I'm going to be focusing mostly on our novel approach, and that combines similarity-based methods and dimension reduction methods, specifically non-negative matrix factorization. And this approach is called SUMO. It's a subtyping using multi-omic data. And I'm going to show you how the SUMO works step by step. But please have it in mind that here we have two main steps. So we are starting from feature matrices, then we are creating similarity matrices, specifically per omics, and then we are working on similarity matrices, which we then create a low rank approximation of. So we're going to use non-negative matrix factorization. So the first step would be calculation of per omics sample similarities. So here we have 3 different feature matrices. They may be of different sizes, different number of samples, different number of features. Let's say that would be, Sumo is specifically an implementation of focusing on continuous data types right now. So let's say that that would be continuous dataset expression, gene expression, miRNA expression, or methylation. And then per each omic, we calculate sample-sample similarities. How do we do that? So we take a feature matrix, and there's some pre-processing that I recommend. That would be a filtering of features that are non-informative, and a data standardization, so each feature have the same weight. And then we calculate sample-sample distances. We're going to use an RBF kernel. This is a radial basis kernel. There is basis function kernel that is focusing on that is taking as an input distances. Here I put Euclidean distances, which is a type of distance that I recommend using for continuous datasets. However, we are implementing alternatives, and there are some distances that may be better suited for your data. For example, there have been research showing that cosine similarity is very good for a single cell sequencing for transposable accessible chromatin, so SC ataxic. So we may use, so if you have this type of data, we would use, for example, cosine similarity. And then we create a distance matrix. And based on the distance matrix, we apply our kernel to calculate a similarity matrices. So here we have n cross n, so there's each sample, and so each column and each row is a distance between a pair of samples. And here, each column and each row, we can point that here, for example, there is a similarity between sample sample I, and sample J. So this is a specific function that we are using. As you can see here, a row is a function of distance. So this we are calculating a distance between 2 samples, and we are normalizing it by average distances between each sample and and this sample nearest neighbors. What is very important to see here is let's focusing on it's to focus on this part, that basically if we figure out what distance is suitable for our data, then we can use this framework to it is very flexible framework, and we can use Sumo to work on our data, no matter what type of data that is. So here there is a very simple example that helped me to understand this at the beginning, how it looks like. So this is a very simple data set. It has only 2 features, feature A and feature B, and there are 2 groups of samples. And here you can see that these 2 groups are clearly separable in terms of feature values, and this is how similarity matrix would look like. So this matrix is sparse. It will have higher values for the higher the values is, the more similar 2 samples are. And there's a 1 on the diagonal. And here what you can see that even though we have some noise here, we can still clearly see that there are 2 clusters just with some amount of noise. So basically, because we are using our kernel, we are dealing with this noise we are dealing with this noise, and we are still able to identify the clusters. So here I am using A instead of, for example, S for similarity. And this is because this similarity matrix is also an adjacency matrix. So if you can think about this as a layer of a multiplex network or graph. So for similarity matrix, so for our graph, we would have each node would be a sample. So for example, here would be a sample in this layer, and each value in our adjacency or similarity matrix would be a weight on the edge between two samples, between two nodes. So here we can see a first attempt to support a missing data, which is if there's one part of missingness. So if there is a missing sample that is not available in our layer, we can still support it. So for example, here, there will just be no connection between this sample and different samples. You can also see that here, it's not a full graph, even between, for example, here, it's not a full graph, even though all of the samples are here. And it may be the case that we have not so many features and we are not able to calculate a meaningful similarity between two samples. But we are handling this just by not putting an edge here. So here that would be a missing value, and we're going to support it later on. All right, so we have a similarity matrices per each omic. So still we have similarities, but we have not achieved the integration. So let's integrate it. So to integrate it, we're going to use non-negative matrix factorization, specifically symmetric non-negative matrix tree factorization, which I know it's a mouthful. So I'm just going to go quickly through a basis of how non-negative matrix factorization works, and how we are expanding on this basic idea. So NMF, non-negative matrix factorization, is an approximation using a low-rank representation. It is very popular in machine learning. It was proposed in 1994 by Patero and Tapper, and then popularized in 1999 by Lee and Sung. And it is popular mostly because it allows for automatic extraction of sparse and meaningful features from the data, and discovery of underlying structure of our data. So what that is, just the basics. So we have a matrix factorization here. So matrix factorization would be, this is a case of a usual optimization case. We have our matrix of origin, so our X here, and we want to find two matrices that when they are multiplied by each other, so they will be as close to our first matrix as possible. So in a classic NMF, we have two matrices. So first matrix would be W, and this is a basics matrix. And we can interpret columns of this matrix as components that build our underlying data, our underlying structure of data. So something that is over and over again in our data points, the basis of our data. And when we multiply this matrix by this matrix H, the cluster indicator matrix, will be as close to this matrix as possible, hopefully. Yeah, and this matrix can be used to, as you can see, this is samples by basic elements. We're using R here because it's also a rank. As I mentioned before, rank in the low rank approximation approaches is most frequently the number of clusters that you want to find in your data. So we try to find R-dimensional approximation of our data, and hopefully our data will be spread on each of the R dimensions, so we can clearly see which cluster is which. So here is a bit of the spin on the NMF, and this is a symmetric NMF. So this is basically a similar case, but here we are multiplying two matrices that are actually the same matrix, just one of them is transposed. And we are starting from a symmetric matrix from the matrix that is n cross n. So I already put A here, so because this is a case in Sumo. So we have our similarity matrix, for example, so n cross n samples, and we are multiplying. We are trying to find a matrix H that, when it's multiplied by the transpose, will be as close to A as possible. So this is just an additional constraint to our NMF. And there are additional curious mathematical qualities here. Basically, you can here think about our data. So on the left, because it's a non-negative matrix factorization, our matrix is non-negative, and because of that, we can take our conclusions from the H. We also have here higher values on the diagonal, and this light blue are lower values, and this will result in the distribution like this in our matrices. You can see that this clearly resembles our similarities that we were looking at before, but it doesn't have to. It will be reorganized as it should. So we extend on this idea. So from general symmetric NMF, we are factorizing. We are doing a three factorization. So we are introducing additional matrix here, and this is a matrix R cross R or K cross K. So number clusters per number clusters, and H was telling her more about where our samples are in our new low rank approximation. So this is a cluster indicator matrix, and here we have a matrix that will tell us more about a relationship between different clusters. So we introduce this. We factorize our matrices, each of our adjacency matrices, in a way that cluster indicator matrix is shared between the layers. So we will have only one cluster indicator matrix for a whole multi-omic data set. So this is the data integration step. And we have separate matrices with specific relationship between clusters for each layer. All right, so this is an example of factorization. So here there are three clusters, and here we have relationship between each cluster, and this is a cluster indicator matrix. In general, we are most frequently, in case of optimizing, of finding trying to find the answer to this problem, we are going to put some additional constraints on our H matrix. So in our case, we put here sparsity. We're using a Frobenius norm, which is very widely used for this type of problems, and we are going to introduce here a sparsity component to make sure that our H matrix is sparse. So we can then look at each row translates to a sample. So we can then look at and apply the maximum value criterion. So find a column which have the highest number for each sample, and this will be the cluster index for this sample. Yes, so this is our cost function that we are minimalizing, and it has a different number. So we may have a different number of samples for each omics. So we are handling that by additional parameter, and then we are introducing an additional matrix that handles missing data inside the similarity matrix, as I mentioned before. So this is an optimization problem. We are using a gradient descent to minimize this function. So you might think about this as starting from a random point, and then we are more or less randomize our matrices. We apply our cost functions and check how close are we to our original matrix. Then we're changing some of the values in our matrices, so we're making a step. And then we are applying our cost functions again. If cost function is the cost is smaller, then we are moving probably in a good direction. There is some stochasticity here, but in general, this is what we are doing. We are trying to find a minimum of our function. All right, so we did a factorization, and we have one cluster indicating matrix. So yes, we have labels for our samples. So that's it, right? Well, not really. The issue here is that, as you can tell, there are some local minimas, and our function is convex in H and S, but it's not jointly convex in both. So, and even if we are starting from not fully randomized starting point, we are going to we still have some randomness here, and we can still land in a local minima. So what we are doing is we are basically we're repeating this multiple times, and we are doing a consensus clustering on a matrix. So I'm going to do a factorization multiple times. I will get labels from each factorization. Then I'm going to create a consensus matrix that shows me how many times two samples were together in the cluster. And then I'm going to cluster this using a spectral clustering to find samples with cluster labels. And then from this, I can go on and identify features in my first data that support our cluster separation. And this is a full Sumo workflow. So our tool is an implementation focusing on this particular cost function that you just saw, and the mostly continuous data sets. But as I mentioned before, the framework is flexible and can be used for different types of data or different cost functions or different similarity measures. It is available on GitHub and on PyPy. It's a Python tool, and it handles fully prepare mode, handles similarity, creation of similarity, so we can not use it and create your own similarities, and then run Sumo on it. So run Vendel's factorization and consensus clustering. We have an interpret mode which implements a tree-based method like GBM to find features from original data that support our cluster separation. And we have additional metrics for comparing different clusterings between each other. Yes, and very important, we also, because we are using consensus clustering, we are able to use a proportion of ambiguous clusterings to decide how to select a rank. So basically, how many clusters is in my data. We are also implementing cognitive correlation coefficient, which is usual for NMF-based methods, but is not so sensitive that sometimes it is very hard to choose. So basically, thanks to consensus clustering, we are able to use a proportion of ambiguous clusterings here. All right. Yeah, so I'm not going to focus on that, but we are, as I show you, we are handling noisy data. We are comparing ourselves with different tools, and we find that we are handling noisy data very well. We're also one of only two tools, to my knowledge, only two tools for biological data handle missing samples in some genomics, and some of these other tools also have an issue that they need imputations. So imputation of original data, which we do not, because we're handling missing data, as I show you. All right. So there is, I don't know how much time is left, but I'm going to show you quickly some practice case study using Sumo. So we have published a preprint right now where we used Sumo to detect relationships between patients in lower grade gliomas. In general, lower grade gliomas, gliomas are neuroepithelial tumors that originate from glial cells. World Health Organization for a very long time used only histological images to classify different types of groups into 4 grades. So we have grade 1, 2, 3, 4, where grade 4 is glioblastoma and grade 1 is astrocytoma. And it was very hard to classify them well, especially for grades 2 and 3. Sometimes the histological images were very similar. In 2016, this classification was revised so that it uses molecular data right now and status of some major mutations that are seen in different types of gliomas. Especially IDH mutations are prevalent in low-grade gliomas. So around 80% of low-grade gliomas has a mutation in IDH1, and 90% of this are this specific IDH1 mutation. In general, if there is an mutation in IDH1 for lower-grade gliomas, the patient has better chances of survival. For grade 2 and 3, after removal of tumor, postoperative radiotherapy remains a standard care. And this is just a quick view to how gliomas are classified. So there was so this is a Secarelli et al. Paper. So this was a study that was the largest study of diffuse grade 2, 3, 4, so no astrocytomas, gliomas to date. And they basically, and based on this, there was a revision of World Health Organization guidelines. They identified that IDH status is the biggest separator between all gliomas. They investigated, I think, around 1,000 samples. And basically they found that IDH status separates samples into 2 clusters. One cluster with IDH mutants contains groups that have a codeletion, groups that, so this is a codal group right here. We also have 2 groups, so we have groups that do not have this particular codeletion that is very frequent. However, they vary in the level of methylation, of genome-wide methylation. So we have either low or high genome-wide methylation. And for wild type, we have classic-like type, which have a classical gene expression signature, mesenchymal-like, which is enriched for mesenchymal subtype tunnels, and there's here there is an PA-like group that's going to be important, and it's enriched for tumors with molecular similarity to grade 1 pilocytic astrocytomas. So they identify that this group has a favorable outcome among the wild type groups of IDH, and this is going to be important. So we looked into it, TCGA LGGs. The data was downloaded from Xena for 556 primary tumors. We looked into continuous data, so RNA-seq methylation and mRNA expression. And our algorithm detects 5 clusters. We can click here, there's a survival curve. We can clearly see that there's a single subtype, group 2, that has significantly worse prognosis. Most of the samples from this group are labeled as classic-like, mesenchymal-like, or CGIMP-low. So without codeletion and with small level of methylation. What is very curious is that in this very significantly different group, there is 18 out of 26 IDH wild-type samples that are labeled as PA-like, but Secarelli, even though based on methylation analysis, Secarelli et al. Identified them as having a more favorable outcome. So what we wanted to see is why we are separating PA-like samples into this low survival group and higher survival groups. So we looked into PA-like samples. And basically here we saw that the Secarelli separation was based on methylation. But if we look, thanks to Sumo integration, we can look at miRNA and gene expression, so RNA-seq, and we can see that for methylation there is no significant difference, but for miRNA and gene expression, we see that these two groups, so groups of PA-like samples that are in subtype group 2 and aren't, are significantly different. All right, and the most interesting part of this analysis is that our subtype 2, so the subtype that had worse analysis, seem to be misclassified. So these samples are clearly enriched for chromosome 7 gain and chromosome 10 loss, which is yet to be used as a marker for separation for WHO guidelines. Even though there was a couple of studies that suggested that it is very important in separation of patients. Here we also see that this particular set is highly enriched for displays a genome-wide low level of methylation. So these two are both hallmarks of that are very specific to GBMs and not really LGGs. Because here we have chromosome 7 gain and 10 loss, we see that, for example, here we have EGFR genes. This is an epidermal growth factor receptor. And because there is a chromosome 7 gain, and this gene is in chromosome 7, we see an amplification here. And in general, somatic aberrations in EGFR are seen in around 60%, 57% of grade 4 gliomas, so GBMs, and are uncommon in LGGs. But here, because of this gain and loss, we see this. We also see that this particular gene that is known tumor suppressor is also enriched here, and this is because it's on chromosome 10. So this is a deletion of known tumor suppressor, which may lead to worse outcome, worse survival. So we looked into it, and pretty clearly we can see that our samples in subtype 2 are much more similar to GBMs than to LGGs. Here I just I'm showing. I hope it is visible. Just a quick view of our analysis, but we also did a lot of additional looks. We used Elmer to look into the hypomethylation more clearly, and to correlate this with transcription factors. But here we downloaded the Excel enrichment scores. These are basically, because we know that tumors are built from a bunch of different cells, and there is a microenvironmental impact here. We are looking into 64 immune and stromal cell types, and their enrichments in TCGA, GBM, LGG cohorts, so LGGs and GBMs together. And here we have it separated by our SUMO clusters, and we can see that the cluster 2 have much more similar silver profile to GBMs than to different clusters. In general, astrocytomas so are assigned to subtype 2, show higher scores compared to other LGGs, NGG subtypes. But they're very similar to GBMs. We also can see that lower methylation and higher regulatory. I hope you can see the colors here. So the tumor assigned to subtype 2 show lower methylation and higher regulatory activity at astrocyte-specific elements. Here, this figure line shows a mean for each subtype, and clearly it's completely different at different subtypes. So basically, what I'm trying to say here is we see here over and over again in different studies, as well as here, as identified by STUMO, that chromosome 7 gain and chromosome 10 loss appears to be a very important marker that could be taken into account for separating patients into different grades. In general, patients that have this particular hallmarks as a subtype 2 may benefit more from aggressive treatments, as the samples seem to be more similar to GBMs, and introduction of this additional criterion will probably lead to reclassifying them as GBMs. So these patients might benefit from more aggressive treatments, and they should be this subtype should be treated separately in clinical trials, as it may answer differently to different treatments, to different drug types.
I'm sorry, we are after time.
Yeah. Yep. And I just finished.
Is very interesting.
Yeah.
And
I just finished.
Slides, it will be nice.
Yep. So this is a marker that we find for methylation probes close to this particular gene, and that's it. So I'm just finished.
Thank you very much. We have time for one very short question. Anna Maria asked, in my experience, feature selection is something that strongly influences the final similarities, even more than the choice of distance metric. Do you do this with SUMO? And did you look how changing feature selection affects the results? Sorry if that was mentioned and I missed it, Anna Maria.
Yeah, so basically we are doing some feature selection, but it's very slight. We are doing some filtering of genes that are mostly non-expressed, and otherwise we are able to, for this particular case study for IGs, we were able to use most of our data. So we just threw away an unexpressed genes that were not expressed in more than 3 or 4 samples, I think. In general, it is possible that it will change the outcome. As I mentioned before, if we can use this framework for different types of data, and for example, we made some we made some experiments and tried to use somatic mutation data. And if we want to use somatic mutation as this is a very noisy and sparse data set, we will benefit greatly from a feature selection in this particular case. So we would want to select only features that are informative to our disease, and then we can use it in Sumo. However, in this particular case, we find that we may be able to do it better if we use a network, some kind of network propagation to, for example, using protein-protein interactions to change our binary data into continuous. And then a feature selection is not needed for this step. So in general, my answer is it's not much needed, but it may change results very slightly and help with stability. Okay. I cannot hear you, Lydia.
Oh, I'm sorry. It's my fault. Thank you, Carolina. I was muted. I'm sorry. Carolina, thank you very much for your presentation. And it's high time for next speaker. The last speaker in the session will be Pamela Miranda from Department on Physics, Federal University of Minas Gerais, Belo Horizonte from Brazil. Pamela will present a presentation how mismatches influence PCR primers in detection of SARS-CoV-2. Pamela is was also a participant of our NGS seminars. We are happy to open your talk. The floor is yours, Pamela.
Okay, thank you, Lydia. Can you hear me?
Yes, I can hear you.
Oh, okay, thank you. Thank you first for the opportunity. I'm very excited with the symposium. So now for the time, now you will start. So I want to present you about our work. We thought when the SARS-CoV-2 pandemic comes, how mismatches influence the detection of this virus, primarily in PCR methods. So some new informations. It's important because just not is the method, but the virus is important to understand something. So the SARS-CoV-2 virus, he affects the severe acute respiratory syndrome of coronavirus 2. It is from beta coronavirus family Coronaviridae. The reference genome has 29,903 base pairs, and its virus is an enveloped positive sense single-strand RNA. So for the PCR detection and other detections like lab, the targets, the region, gene regions, we are extract from the genome. So some conservative regions mainly are the N gene, the E gene, and S gene, like protein. So all these gene regions are the targets for detection of SARS-CoV-2. So from these regions, the primers and probes for PCR are designed. So PCR is the polymerase chain reaction, and there is many variations from it. RT-PCR, multiplex PCR, and digital PCR, for example. For SARS-CoV is interesting. The process with reverse transcription, because of the virus has its genetic material as an RNA. So RNA degrades fast. So it's necessary to have a DNA template for the process of PCR. So the sample has RNA material, the reverse transcriptions acts to obtain the cDNA. So we have the version in DNA of the virus. So from this, it's the main idea of PCR in a simple way. So how it works is you have first a double strand. You can imagine the RNA and DNA template. So in the first stage, you denature this double strand, you separate them. So from one double strand, you obtain two single strands. And it is in a high temperature to break the hydrogen bonds that bond the base pairs from each strand. Second stage, we have the annealing. So it's the first time the primes come and hybridize with each single strand. And it's a peculiar temperature because it's the moment you need very, very attention. And the last one is the extending stage when the DNA polymerase comes and elongates the primers and obtain the amplification, the amplicon of the target. Okay, so these three stages make a cycle, and PCR methods do from 30 to 40 cycles. Okay, so as I said, anneal stage is very important. Each stage is very important, but one of the parameters is very, very careful to do is the design of primers and probes for PCR, because the anneal stage is the point when the primers will hybridize. And if primers don't hybridize in a very good way to the template, the amplification not occurred. We can't say if is the SARS-CoV-2 in this case or not. Oh, okay. So for this, a parameter, important parameter is the melting temperature, because this stage is dependent from melting temperature. The temperature when half of the DNA solution is hybridized and not. Okay, so for this, what you need, we intended to calculate, to predict the melting temperature for all primers and probes we collected. For this, we used a simple and interesting model called the Peyrat-Bishop mesoscope model. This is a statistical thermodynamics model, and it's very simple because he's measured the DNA sequence in a bidimensional plane, and it's cheap for computational working. So here we have the molecule, DNA molecule. So in left strand, one strand, and the right another strand, and each colored point is a base, nitrogen base. So what is the idea? Between bases in two strands, we have the hydrogen bonds. Bishop interpret this like a Morse potential. Okay, so we have a potential for the hydrogen bonds, and the same is for the interaction, the stack interaction between bases in one strand. and it's interpreted by like a elastic potential. Okay, what is interesting? Because the model considers separated this potential. Okay, so it's independent. And after this is related in a Hamiltonian. So we have second point. Okay, here we have the kinetic energy. In the second, these two W, little w, are the stacking interaction. And the last one is the most potential, like I showed. So from this, we can obtain this tau. This little tau is an index, a statistical index for each prime and for each group. And with these little a's, they are the regression coefficients, we can predict the TM, the melt temperature for each prime and probe. Okay? But each prime and probe, we align this. I will explain for you. So first, our collection, our data set. First, I forgot to say, for this calculated, because we are considering mismatch, we use the parameters that we parameterize for all sorts of mismatch, more than 4,000 sequences with containing up to 3 consecutive mismatches. It's early work. Our data set, we collected 92 published sets. So we have sets from WHO, CDC, Charité, and other groups. We are collected from NCBI 7,389 genomes of SARS-CoV-2 and another 323 genomes of other 6 types of coronavirus. It was our control group because we want to see if the prime samples we collected can detect only SARS-CoV-2 or maybe have some cross-react. So this is our workflow. So we collected the primes and probe sets, the genomes, and we aligned all of them. So all of our sets are aligned with all SARS-CoV-2 genomes and other coronavirus genomes. And with the alignments, we predict the melt temperatures. So we calculated these temperatures for the interaction, the hybridization of the genome and primer, because it's interfere in what I said before, the anneal stage of PCR, sorry. Because it's important that the prime and probe hybridize in a very well way with the genome, with the target, okay? And after this, with the melting temperatures, we can calculate the coverages for all primes and probes. How we did it? We restricted the genomes more than 25,000 base pairs. We considered just 1, 2, or 3 mismatches, because more than this, the melting temperature is substantially, so it's not good to use this situation. And we range the temperature in 5 degrees, okay? And calculate the coverages. The first coverage is for cases with perfect match, when we have no mismatch, just a perfect situation. So we considered the total of genomes above 25,000. So we had 7,016 no alignments, because the prime samples, there are situations they are not aligned with the genomes. And we out the mismatch situations, this coverage. And we have another one type, coverage is for mismatch case. It's the point. So again, the genomes not aligned, and this this number, when we consider this range, we have the melting temperature. We turn all of the prime samples on double strand, calculate melting temperature for them. It was our reference temperature. It's a similar situation for perfect match. And with this range, we can restrict situations in the mismatch presence doesn't contribute to our coverage, because a law of 5 degrees in relation to reference temperature, it's not good for PCR method. Okay, I hope so. Zoops, because we collected 340 primes and probes, so we have much ambiguous, so very much results. But here I bring for you to see a set of CDC, the N1 set. So we have the forward prime, the probe, and the reverse prime. This is the TM, because we analyze if the sets follow the recommendation of the prime and probe design for PCR. And here the coverage for prime and the coverage for perfect match. So we can see this set of CDC is good, is covers a good number of genomes, perfect way. And when we consider the mismatch presence, it's changed a little. For the forward prime, it's not the difference. The number of mismatch presence is not considered. It's probably, as I look at, it's decrease the temperature. So this number is the further times with mismatch we consider. So 0. But when we look to probe, we see a increase in coverage, and the presence of mismatch contribute with 166 occurrences more. And in this case, for reverse, we have a little degree too, so 20 more. Okay, so it's only considering the just one group, because for the PCR method, in general, we design a pair of primers, so the forward and reverse, and the probe. So for the CDC example, I bring it. But we see it in other cases. For example, to group, we have increases, interesting, 96% with perfect match to 99% with mismatch presence. And so we consider further 168 alignments. It's very, very interesting. For Davida et al., I bring the pair prime or prime pair. We have the forward and the reverse, and just 7 and 2 more are a little increase. Okay, and it's interesting because the viral RNA virus have mutation rates, and in case of the SARS-CoV-2, we see different genomes in our world. So in case in an urgent situation we are, mismatch maybe be considered to contribute for the detection. But it's very good, but we've observed a special case. It was for RDRP primary probes. In the case of Coleman et al. From Charité group, for the reverse types of primes, we obtained 0 for all situations. So these reverse primes don't align in perfect way with any genome, and in this case, more mismatch presence don't contribute. The same for the primary pair of the new et al. 0 for all, not perfect, not mismatch. It's different for this forward prime of Coleman et al. Because it has about 8 or about 0% with perfect match. The method about 2 or 3 genomes, but when you consider the mismatch presence, it's increasing crazily for 99%. So in this case, we have 6,991 more. So although this not perfect way with genomes, it can be detect the SARS-CoV-2, which are methods of diagnosis. Okay, all my time. So some conclusions is we perceive in a few cases the presence of mismatch decreases significantly the temperature of primes and probes, but in some cases we have considerable situations with the mismatch presence contribute to that detection. In some cases, like we saw about the forward prime for IDRP from Coleman et al., it's 0 on for perfect match to 99% considering mismatch case. And our perspectives of this work is analyze future prime probe sets because it's our design, have designed continuously. We want to optimize the prime probe design with our parameters and our data, and to extend this analysis for lamp primes too. I want to thank NGSchool, Symposium, and all the organizers. I follow you, and the pandemic is good because I meet you. Thank you for the opportunity. I'm very happy. Acknowledge my financial support to my university, UFMG, and my department of physics, because I am physicist and my supervisor too, so we work at physics department, bioinformatics program, which I am part, my master degree and my PhD course now in bioinformatics. Oh my God. My lab, this is my lab, my colleagues and friends. This is my supervisor, Professor Gerald Weber. So we are in English, computational biophysics group. If someone wants to be in touch, my email, okay. Thank you and be safe and hope for the best because we will pass for the situation. Thank you.
Thank you very much, Pamela. It was a very nice presentation. And we would like to thank you too. Very nice for us to have your talk. And you have 2 questions. I have 2 questions for you. First one, does the location of the mismatch have an effect of alignment?
Yes, it's the next step, because when you have the mismatch near the pre-end of the primer, it may be difficult, but the presence of the DNA polymerase. So it's difficult to amplify the amplicon, the target. But in our case, first we analyze the presence in all positions, because it's only the first goal to see the hybridization with the template.
Okay, thank you. And second question, do the 19 PCR primer sets have the same PCR target? If not, what is the most common target for SARS-CoV-2?
Oh, the common for SARS-CoV-2 is this, just a second. This I bring to you is the mainly so spike, envelope, nucleocapsid, and RDFP, but I read this there is some problem. But this is the first, because this is a conservative regions. It's not the mismatch, the mutations in these regions are small rate to mutate.
Okay. Thank you very much, Pamela. And thank you once more, all of the speakers of this session. It was last talk of the session. And now we have some short break. During the short coffee break, you can join sessions discussion panel on Discord or ask questions on YouTube. And now, and we will have second session at 5 p.m. Central Europe time. And now grab your tea or coffee and see you in more than 10 minutes. Bye-bye.
Hello, everyone. Welcome after the break. My name is Natalia Strong, and we'll start very, very shortly. We just run into some slight trouble with the screen sharing, but it's going to be resolved in a few moments, so stay tuned for the second part of the conference. Hey, Vitalina, if you would please share your screen, that would be great. And OBS. Hello again, everyone. It seems we are ready, so it's my great pleasure. Once again, my name is Natalia Strong, and once again, it's my great pleasure to invite you to the second session of our NGSymposium. So our first speaker in the second session is Vitalina Bashinska. Welcome. Vitalina is from a Diogen Genetic Laboratory in Kyiv, Ukraine, and her talk is entitled setup of gene reader NGS system for clinical research of cancers. So, Vitalina, the floor is yours, and welcome everyone.
Yes. So, do you see my screen? Hello?
Yes, we see your screen.
Oh, that's great. So, dear colleagues, so is it fine? Just hear me.
Yes, it's fine.
Oh.
You can go.
Thank you for inviting me here to participate in this symposium. I'm not a bioinformaticist. I'm a wet lab biologist with some interest. Sorry. So since something has gone wrong, dear colleagues, thank you for inviting me to participate in the symposium. I am a wet lab biologist with minimal experience in bioinformatics and with some experience in biostatistics, but I will be happy to present you my work on my experience on setup of gene reader next generation sequencing system for cancer clinical research in our laboratory in Ukraine. So the issue is that tumors are really very heterogenic. They contain some set of primary mutations, which are at the beginning of clonal expansion, and additional mutations that occur during the life cycle of tumor. They can occur in a single cell, maybe stem cell, and they can give some advantages of to such cells so that they expand, and so that if we cut the tumor at different points of time, we will get different patterns of heterogenic cells and of heterogenic mutations and of mutations, which is so called tumor metanome. And we can see it if we perform a high throughput sequencing of the tumor, that there are a number of reads with different mutations. And in some cases, we can use this knowledge because some mutations may play a significant role in therapeutic resistance or therapeutic response of tumors, so that if there is a significant fraction of tumor-resistant cells or treatment-resistant cells, they will survive after treatment and expand and give beginning to new labs. So in some cases, if we know about existence of some mutations, it can be useful in clinical practice. For example, there are certain mutations that make tumors either sensitive or resistant to EGFR tyrosine kinase inhibitors, and as a rule, the mutations occur not in every gene, but in a certain set of genes. So all types of genome sequencing are actually used in oncology. Whole genome sequencing is good because it allows to know about every type of mutations. It has not very high sequencing depth, however, but it can allow us to search any mutations throughout the whole genome. Whole genome sequencing is more cost-effective. It gives more sequencing depth, and it also allows to identify all kinds of variants, but within exome. And targeted sequencing is quite frequently used in practice because it allows to reach very high sequencing depth. It's really not more than it's really much more than 500 times. It's actually several thousand times reads of regions of interest.
Hello.
Yes.
Sorry,
Hello.
sorry. I think you're sharing the wrong screen because your slides aren't moving and are not in full screen. No worries.
Sorry,
no worries.
I didn't hear you.
I think you're sharing the wrong screen because your slides aren't moving and you're not in full screen. Don't worry. Just put your screen in full screen mode and we'll see your slides fine.
So how should I change
I think
the screen?
it's F5. I think it's either F5 or on below you have like those 4 screen modes, like right beside the screen size, like the plus minus in the lower corner on the right, and the last one is you would put it in presenter mode. Or try F5.
I really don't understand. What should I do because of settings?
No, when you're in your PowerPoint, like on the bar down, you've got the 4 little screens. Like you have the percentage on the bottom right, and
In the Ninja? Where?
in your PowerPoint,
Oh, in PowerPoint? So when
yeah,
I share my this screen, you don't see the whole screen, right?
yeah.
And I don't see the message. So there was something about share another.
You know what? Maybe just don't worry about it. Just stay on this screen, but change your slides because we haven't seen your slides, and that should do the work.
Mm-hmm.
Because there we see your slides, and this is much better.
So I can share the whole screen. So I will do, and sorry for this because I'm really not strong.
Don't worry about it.
Can I do? Can try to do this once more. So I'll go to sharing my screen.
Try maybe Alt Enter. There was a tip like that. Maybe this would help.
So will this actually work? Do you see my whole screen? It's like I have some technical problems. Do you hear me?
Yes.
So do you see my whole screen?
Yes. Sorry for that, everyone.
Right, you see my whole screen.
Yeah, now it's perfect. Thank you very much, and sorry for the disruption.
Oh, so sorry because I didn't do this correctly the first time. Targeted sequencing with regions of interest covered is maybe the best choice for the maybe not maybe for primary screening of tumor when you just deal with molecular pathology, and you can also use RNA sequencing, whole transcriptome or targeted sequencing. For example, you can detect RNA fusions in non-small cell lung cancer, and single cell RNA-seq is also really great, but it's not applicable for the majority of clinical cases when you just need to provide some kind of analysis. What we have done here was set up of gene reader sequencing system, which uses targeted sequencing. Specific regions of interest are enriched using targeted PCR, which are then sequenced mapped to the reference genome, and panels are developed such that targeted variants are of non-actionability, but certainly we read the certain exons of certain genes, so that we detect any variants which are present there. This system looks like this. It is really developed more for clinical laboratories, so that you have options to automatize all the stages, nucleotide collection, extraction, library preparation, next generation sequencing, and there are solutions for data analysis and interpretation, and also solutions for quality control, which are maybe quite standard for next generation sequencing technologies, because you need fluorometry, spectrophotometry for quality control DNA, and for libraries, and capillary electrophoresis for also control of libraries. There are different input types for these panels and for oncology sequencing overall. They include FFPE samples obtained during biopsy. They allow to preserve DNA samples for years, but they have several difficulties because formalin fixation leads to DNA strand breakage and to cross-links between different proteins, between different DNA strands. There are also possible cytosine deaminations into deoxyribosyl that lead to sequencing artifacts, so C to T or G to A. And what the used chemistry uses is uracil and glycosylase that converts at least the majority of such artifacts, any cytosine deaminations, and it is adapted to highly degraded DNA to some extent. Another type is plasma, which represents liquid biopsy, and some panels of QIAGEN generator are adapted to such technology. It is good because it doesn't have such artifacts as formalin fixation, but also it has very low yields of circulating tumor DNA, and it can also contain artifacts like contamination with genomic DNA. Whole blood, bone marrow are also used for different blood cancers, and certainly you can also use fresh frozen samples if you have ability to freeze the biopsies soon after they have been taken. And several quality controls are also used. We use fluorometer to look at concentrations of DNA or RNA, purity, which is assessed with spectrophotometry, and different integrity assessments and amounts from the file DNA, which is assessed, for example, with multiplex PCR. So this is a brief introduction to panels. For ovarian breast cancer, we can use FFPE or fresh frozen sample. Regions and exon intron boundaries, and we can also use other panels which are adapted for all solid cancers and contain most common driver genes. There are also panels for non-small cell lung cancer, which assess mutations in DNA and in RNA and in RNA fusions, and they can be combined so that you can get the more information, which can be clinical relevant, and you can also give some clinical which is basically a set of aqueous droplets
Vitalina, we cannot hear you and neither we cannot see your screen. Please try to say something and to share your screen on OBS.
Hi, anyone hear me? Hello, anyone can hear me?
Hello, I can hear you.
Okay.
Yes, we're hearing you, Lucia. Just give us a second.
This is my presentation. Can you see it?
Please wait a moment, Lucia. Natalia will introduce you in a moment.
Okay, I'm sorry.
Yeah, no worries. So it seems that Vitalina has some connection issues, I would say. She disconnected quite abruptly. So if you have any questions to Vitalina, we will take them on Discord and pass them to her. And we hope she will answer them as soon as possible. But nevertheless, we have a next speaker that's ready. So I am very sorry if I butcher your name. I should have asked before. So we have Lucia Hawk, and she's from Croatia, from Computational Organic Chemistry and Biochemistry Group. And she will present her talk, which is entitled "And Substitute Benzimidazole as Tubulin Polymerization Inhibitors." That's a mouthful, but it seems like a great talk. So Lucia, the floor is yours, and go ahead.
Thank you for your kind introduction. Can you hear me well?
Yes, and we see your screen, however, not on full screen mode.
Not full screen.
Yes.
Okay, just a second. mm, can you see it now? Hi.
Not full screen yet.
I don't know what to do. I can see that.
Shimon?
And now I have to be in my window for my presentation window, or yes, I did this, but obviously it's not. Okay. I don't know what. How can I do this? I will try then.
Vitalina, I wrote you a private message on Discord if you would like to look at that, please.
Is it okay now? Can you see the whole screen? I have to be in my presentation on my computer, which I choose to share, or I should be at this tab in internet. Where should I have to be? In which tab I have to be? In my presentation, my PPT, or in this window on the internet? Yes. Mm-hmm. Yes, and when I in my, is it better now? Can you see the second slide? No, nothing happens. You can see when I scroll it, but you can't see when I change to full screen. You can see this change now. You can see the full screen of fourth slide. But it's fourth slide. What do you mean about proceeding with presentation in not full screen mode? Is it okay? I don't have any animations, so I think this would be also fine. Okay. Can I start? Everyone can hear me very well.
Yes, we can hear you.
Unfortunately, I'm not able to share the full screen with you, but if you are okay with it, I will procedure with this half screen presentation.
Go ahead.
Okay. Thank you for your kind introduction. I am Lucia Hock, you said very well, and I am a PhD student in the computational organic chemistry and biochemistry group at Ruđer Bošković Institute in Zagreb, Croatia. And I will talk you about 10 substituted benzimidazole acrylonitriles as in vitro tubulin polymerization inhibitors. I would like at the beginning to thank the organizers for your for the accepting my abstract and giving me an opportunity to share with you some interesting results. And I would also thank to the all members of my multidisciplinary team for very nice and successful collaboration. Benzimidazoles have recently become an important subject of intensive investigations in organic and medicinal chemistry due to their interesting chemical, biological, and pharmacological features. Benzimidazole scaffold as a small nitrogen containing heterocycle represents a fundamental building block widely incorporated in the structure of numerous natural and synthetic molecules. The fact that its nucleus acts as a bioisoster of naturally occurring purine is of great importance since benzimidazole derivatives play a crucial role in the function of many biologically important molecules as a result of the interaction with DNA, RNA, or different proteins. Previous results showed that benzimidazoles related to 2,3-desubstituted acrylonitriles exerted pronounced antiproliferative activity, particularly in the case of N-derivatization. Such systems displayed high selectivity in HeLa cells with IC50 values between 0.8 and 0.7 micromolar. Mechanism evaluation of the anti-tumor activity of the structurally similar systems, like presented triazolopyridine substituted acrylonitrile, showed that such compounds have influence on the tubulin polymerization. Based on these findings, we designed the novel N-substituted 2-benzimidazole acrylonitriles and evaluated their antiproliferative activity towards 8 human cancers and 1 non-cancerous cell line, and to explain the mechanism of the biological action, the tubulin polymerization assays were carried out on the most promising agents. Additionally, to give a deeper insight into binding within colchicine binding site in tubulin, those molecules were computationally analyzed. Orthophenylene diamines were prepared by the uncatalyzed microwave-assisted amination, starting from the corresponding orthochloronitrobenzenes and followed by the reduction of the compounds from 3 to 6. And substituted 2-cyanomethyl benzimidazoles from 13 to 18 were obtained in the cyclocondensation reaction with 2-cyanoacetamide. Finally, and substituted 2-benzimidazole acrylonitriles were synthesized in condensation reaction with compounds from 30 to 18 with chosen aromatic aldehyde. All acrylonitrile derivatives were obtained as E isomers, except 30, which was a mixture of E and Z isomers with 5:1 ratio. All newly prepared compounds were tested for their antiproliferative activity in vitro, as mentioned before, on 8 human cancers and 1 non-cancerous cell lines, using staurosporine and docetaxel as references. Most of them exerted a moderate activity and did not show any significant selectivity towards the tested cell lines. Compounds 30 and 41, both bearing isobutyl side chain on benzimidazole nucleus and having the NN-dimethyl amino substituted phenyl ring, exhibited strong and selective antiproliferative activity in sub-micromolar range of IC50 concentrations, but unfortunately were less active than the standard drugs. However, all tested systems were significantly less toxic against non-cancerous cell in comparison to reference staurosporine and docetaxel. To obtain an insight into the molecular mechanism of action, the most promising compounds 30 and 41 were tested for their in vitro ability to interfere with tubulin polymerization process. It turned out that these lead compounds cause disintegration of the microtubule network comparable with the effect of microtubule destabilizing reference compound vincristine, and in contrast to the microtubule stabilizing reference compound docetaxel. Computational analysis was performed to offer more insight into binding of the tested systems 30 and 41, and for that purpose we employed the docking simulations and quantum chemical calculations with the aim of obtaining binding poses and accompanying binding energies. Given that microtubule destabilizing agents typically bind either to the vinca alkaloid site between two longitudinally aligned tubulin dimers, or to the colchicine sign at the intra-subunit interface within a tubulin dimer, we decided to also include colchicine in the analysis as a reference system due to its better structural similarity with our ligands. The predicted binding position of colchicine very closely matches to that from the crystal structure, suggesting that the docking procedure position it with the colchicine binding site as shown on the right figure. Calculated binding Gibbs free energies for all studied compounds are given in the table, and the analysis was also utilized on structurally similar but biologically less active 29 and 42 compounds. It turns out that the highest affinity is predicted for colchicine with binding energy minus 9.3 kilocalories per mole, which is in excellent agreement with experimental volume. Compounds 41 and 29 possesses identical binding affinities, but very important to emphasize at different binding positions. Namely, all compounds except 29 most favorably bind within a colchicine binding site, and 29 binds on the other end of tubulin beta subunit. The most favorable position for 29 within the colchicine binding site is 0.5 kilocalories per mole less exergonic. The highest binding affinities as well as the correct position within a colchicine binding site for compounds 30 and 41 could be responsible for their antiproliferative activity. The observed differences between 29 and, for example, 30, which only differ in presence of NN-dimethyl amino group, can be explained in the following way. NN-dimethyl amino group possesses a strong electron donating character, and its donation of electronic density primarily into the neighboring phenyl moiety is significant for the binding. Within the binding site, compound 30 is predominantly stabilized through cation P interactions between phenyl and lysine 352, which clearly benefit from its increased electron density related to NN-dimethyl amino group. The lack of this structural element in 29 results in its differing binding position within colchicine binding site which is rotated to position its benzimidazole unit in the favorable cation P interactions with lysine 352, while the phenyl group interacts with hydrophobic amino acids, and the cyano group form hydrogen bond with cysteine 241. In 30, the mentioned hydrophobic pocket is occupied by the benzimidazole fragment, allowing the cyano group to form hydrogen bonds with lysine 254 and asparagine 258. Cysteine 241 would likely interact with compound 30 either through hydrogen bond interactions or SHP interactions with benzimidazole unit. System 41 features additional cyano group on benzimidazole core, which makes it 0.3 kilocalories per more weaker binder than 30. Unlike dimethyl amino group, the cyano group is a strong electron donating, electron accepting moiety, thus reducing benzimidazole's potential to form a pairing with cysteine 241, like it happens with compound 30. This lack of interaction is only moderately compensated by hydrogen bond of the introduced cyano group with asparagine 249. Lysine 352 now interacts with central cyano moiety, which is clearly a weaker contribution. Lysine 254 again forms hydrogen bonds with benzimidazole nitrogen bearing isobutyl group. Replacement of the N-isobutyl group in 41 with smaller N-methyl group in 42 results in rotated position of this compound in the active site and in reduced affinity by 0.2 kilocalories per mole, and its benzimidazole unit bearing cyano group interacts with lysine 352 through cation P interactions, while its N-dimethyl amino group form hydrogen bonds with cysteine 241. We hope that this computational analysis will allow some enough structural and electronic information to offer some general conclusion about tested compounds and to aid in a design of more or even more potent ligands based on the employed organic framework. Biological assessments of compound 30 was carried out on the mixture of E and Z isomers, since they could not be efficiently separated by column chromatography. So we decided to analyze the influence of the isomerization around the central CC double bond on observed affinity. Our DFT calculations confirm higher stability in the E isomer, and the docking results show that Z isomer also binds in the same Colchester binding site as the E isomer, yet with a slightly lower binding energy. Given that, we can conclude that E-isomer is the likely active compound in performed experiments. To conclude, we have designed, synthesized, and biologically evaluated novel and substituted benzimidazole-based acrylonitriles, and two of them, 30 and 41, displayed selective antiproliferative activity in sub-micromolar range of concentrations. Further mechanistic studies demonstrated that this class of compounds inhibits cancer cell proliferation by disintegrating microtubules. Computational analysis confirmed that binding with the colchicine binding site is governed by interaction of phenyl unit with lysine 352, which is promoted by NN-dimethyl amino group. Hydrophobic interactions with N-isobutyl group ensure a proper ligand orientation within colchicine binding site. At the end, I would like to thank thank you for your attention, and if there are any questions, feel free to ask. I will finish my presentation with this beautiful picture of Zagreb. Hello?
It is really a beautiful picture of Zagreb. I have not visited. I have, however, a question to you, if you don't mind.
Yes.
So, if I followed and if I understood you correctly, you identified the network, the first one, that is important through lab experiments. Is that correct? So then you went to computational analysis. And do you think it's possible to do it the other way around, like find a network through computation and then verify it in the lab?
Yes, computational analysis would actually, it would be more correctly if they are performed before experiments. But here we use the computational calculations to identify the binding site of molecules. Through experiments, experiments showed that mechanism of action is destabilizing microtubule network, but there are more sites. Different agents can bind to different sites of tubulin to cause disintegration of microtubule network. For example, zinc alkaloid site and colchicine site. So we performed docking simulations to identify which binding site are the most proper for these molecules. And
Okay.
we identified that that there is colchicine binding site which is more proper for these molecules.
Okay, thank you. And another one. So based on the results of your analysis, do you think the anti-tumor activity of your compound, the N-substituted benzimidazole, is tumor specific, or is it just should I say, is it tumor specific or cancer specific? I mean, if it's just one type of cancer of its parent cancer. I don't know if that's clear.
Well, I think for that conclusion, we should run some experiments, animal experiments, or some other kind of experiments. But I think that these compounds will only serve as some precursors, some pre-compounds to to further investigate, to further try to improve their activities, specifically to some cell lines, to specific cell line. Maybe in this direction, the next phase of research will go. So the additional synthesis of more potent ligands.
Okay, thank you very much. Thank you very much, Lucia. It was a great presentation.
Thank you.
And thank you very much. And I think we are ready for our next and last speaker in this session. So we have a great pleasure of presenting Agata Rakshevska, who is a postdoctoral researcher fellow at Charité in Berlin. She did her PhD on single-cell transcriptomics using microfluidics, but what she will present today is slightly different. Because today she will present the consortium EasyGenomics, which stands for European Advanced Infrastructure for Innovative Genomics. So, Agata, thank you very much for joining us, and the floor is yours.
So, hello, everybody. I would like to start from stating that I'm really glad that I have the opportunity to contribute to this event. I really like what NGSchool is doing, and I followed some of your Thursdays workshops, and I really enjoyed them. But as you mentioned before, today I'm here to present EasyGenomics, which is a European project which started last year. I joined the project beginning of this year, and as you mentioned before, I'm affiliated with Charité in Berlin. But let's start from what's the mission of the consortium. And our main mission is to provide easy and seamless access to cutting-edge DNA sequencing technologies to researchers not only from academia, but also from industry. So all of that, of course, within a framework that ensures compliance with ethical and legal requirements, as well as fair and secure data management. But that's not all. We also aim to build a community of practice that leverages advanced sequencing technologies beyond country and sector borders to tackle global challenges in science. So how do we do that? So EasyGenomics unites 16 partners across Europe. That includes 12 of the biggest genomics facilities in Europe. The budget of the project is 10 million euros. From that, 6 million euros is dedicated to transnational access calls. I will discuss this in a later part of my presentation. The project will last for 4 years, and it will include 4 calls, and we will fund approximately 150 projects. But the transnational access is not all EasyGenomics do. We also develop new NGS-based methods. We work on benchmarking and harmonization. We also research our users' need to be able to better tailor our offer to those needs. So if you want to help with that, I would like to invite you to fill out a survey on our website. Maybe I will later post a link to it in Discord. And also we keep our users up to date with the latest technologies via a newsletter. But as I promised, let's move now to transnational access. So EasyGenomics provides transnational access through 12 state-of-the-art genomics facilities I mentioned before. So EasyGenomics facilities are spread across Europe. So we have one facility from Barcelona, one from France, one from Kiel in Germany. We have 2 facilities in Berlin, one from MDC, the other one from Charité. We have a facility in Leuven, 3 facilities of SciLife Lab in Sweden, 2 from the KFZ in Germany, and 1 in Estonia in Tartu. So the researchers who are interested in benefiting from this free of charge services EasyGenomics is providing need to apply in one of our calls. So those calls have a couple of rules. One of the most important ones is that access is transnational only, as the name would suggest, which means that applicants cannot request access to services which are only offered by facilities from their home country. And here I would also like to mention that we not only offer sequencing services, but we support our users from experimental design through sequencing, but also we support them with data analysis. So we offer 28 cutting-edge genomics services. To name some of them, it's advanced epigenetic sequencing, challenging DNA sequencing that includes, for instance, ancient DNA and FFPE, immunogenomics, in situ sequencing, long-read sequencing, metagenomics, single-cell sequencing, and as I mentioned before, we also offer integrative data analysis. So, as I said before, the researchers who would like to benefit from these free services need to apply in need to submit proposals to one of our transnational access calls. Those proposals are externally evaluated by reviewers, and this way we can make sure that the best projects are selected. So where are we right now in this process? So we are approaching we are soon approaching the middle of the lifetime of the project. So 2 first calls are closed and the projects are being carried out. In the meantime, we also had an extraordinary call for access related to COVID-19 pandemic. But there are more calls coming, and the dates will be announced on our website and social media. So if you're interested, please follow that. So let me discuss now more in more detail the past calls. So the first call was in April 2019, and the topics that were offered included ancient DNA, single-cell sequencing, metagenomics, epigenetics in human diseases, DNA and chromatin alterations, and de novo assembly. We received 90 proposals, and 32 projects were selected, which means that the success rate was around 35%. So in this call, the proposals came from EU and Horizon 2020 associated countries, and all of them were from academia. So in the revision process, the criteria was impact and excellence. In this call, the most proposals we received from France and from Germany. The second call took place the same year. Some topics were the same, like ancient DNA, single-cell sequencing, or epigenetics in human diseases. There were also some new topics like spatial transcriptomics, long-read transcriptomics with PacBio, or long-read sequencing with Oxford Nanopore. This call was worldwide, and we received 60 proposals, out of which 33 were selected, which means that the success rate was around 55%. So this call had the most proposals from Spain and from UK. As I mentioned before, we also had a special COVID-19 call. That was our response to the COVID-19 crisis. And this call was special on many different levels. So first of all, this time the transnational rule was lifted. And the call was focused and was only internally reviewed, and it was a technical review. So we received 9 proposals, and 7 of them were funded. That resulted in an exceptionally high success rate of 78%. So some more statistics on all of those calls taken together. So if you look at this map, the most proposals we received from Spain and from Germany. And if you have a look on the eastern and southern part of this map, you see it's a bit gray. So, I mean, of course, we are not going to favor anybody, but we would really like to see in next calls a bit more applications from those countries. I hope maybe this presentation can help with that. And so if you have a project which you think may benefit from using some of the latest state-of-the-art NGS sequencing technologies, and you don't have access to those technologies, I would really like to encourage you to submit your proposal in a next call. So as I said before, the next call is coming somewhere in 2021. So to stay up to date, please follow our Twitter and our website. And if you have some questions, you can ask them to me right now or later via email or Discord. Don't hesitate also to contact other partners of Easy Genomics. We're happy to answer your questions already during the phase of preparation of the proposal. Thank you very much, and I hope there are some questions.
Thank you very much, Agata. You actually answered most of the questions I had during the presentation. There's one question from YouTube. Someone is asking, are you hiring right now?
Not that I know of. I mean, maybe some of the partners are hiring, but I don't have this sort of...
I think they're going to be very sad. I did have a question. About the next call, or actually all of the calls, all of them. So how would a typical proposal look like? Do they have to be, because I imagine you explained that you have to have a project that would benefit, but what are some of the criteria you need to pass to be awarded the project? Like a general rule book, what would help you in getting your project accepted?
I mean, all the rules are summarized on the website, but in general, the deciding factor is, as I mentioned, is excellence and impact. So we try to, I mean, that's the reviewer's decision, but that's what they keep in mind why scoring those proposals. And as I said, there are a couple of rules. So the technologies need to be offered by facilities from different country than you are applying from. And for instance, if somebody is granted a proposal, if somebody's proposal is selected, then until the sequencing and data analysis of and that proposal is finished, they cannot apply once again. But you can, for instance, if your proposal hasn't been selected in one of the call, you can reapply next call. But I mean, one more important criteria is that the proposals need to be feasible, so it needs to be possible to be carried out.
Okay. So do you accept like shorter term projects for PhD projects, for example, or is there a time limit even to the realization of the project?
Yeah, so the realization needs to finish by the end of the lifetime of the project. So of the whole Easy Genomics, which is in 2 years. So actually short projects are perfect. It's even, that even more fits with our goals. And so if you really have something in mind, which is just a small part of your bigger project, but it would, be nice to use the technology, which you maybe don't have, that's really what it's meant for.
That's awesome. So the last question from me, I think. So is the more a technical one. So the number of calls from different countries, is it correlating with the number of partnering institutions across Europe in any way, or it's just...
Yeah, I mean, I haven't had a look into that, but I mean, maybe, but yeah, we definitely encourage applications from countries where we don't have partners. So the country is not, yeah, it doesn't matter when the application is being evaluated.
I see.
And there are also some rules that, for instance, you know, the PIs involved in the easy genomics cannot apply, of course, and so on. So there are some limitations. Of course, we cannot apply ourselves for those goals. Yeah, but all the rules are summarized on the website, and if something is missing, let me know. If I don't know the answer, I will try to also figure it out. I'm a part of Technology Watch and implementation group within Easy Genomics, actually. So I personally don't deal with the transnational access, but
So great, thank you. We will link the page to your project probably on our social media soon, so everybody can get a hold of you. And if anybody has any question to Agata, I think you said you would answer on Discord or in email. So we invite you all to talk to Agata and send your project to Easy Genomics. So thank you very much, Agata. Thank you. So this was the last talk in our second session. We would like to invite you all tomorrow at 3 p.m. Eastern set Warsaw time for the third session, the second day. We have 2 sessions tomorrow. And also we would like to ask you, please, if you would fill our feedback form. It's on YouTube, it's on Discord. It would really help us a lot for organizing future events. And thank you again, everybody who presented today. All the presentations were great, even if they were a little interrupted. We are working on a solution for Vitalina's presentation, so you can see it anyway. And thank you everybody who sent an abstract and attended today's conference. Thank you all, and see you tomorrow at 3.